most fundamental level, I'm a basic scientist interested in the neurochemistry of the brain and how that chemistry is related to behaviors that allow us to survive effectively in the world, and also what happens when that chemistry gets distorted and leads to abnormal behavior and thoughts and feelings. So I'm looking for the neurochemical bases of behavior. Recently, we've become very, very interested in the interactions between single nerves, okay, and that's called a synapse. So every brain, your brain has about up to 10 billion individual neurons, most of which are separate from one another. Tiny little gap separates one from the other, and those points of contact are called synapses. Information moves across these gaps in the form of small chemical molecules called neurotransmitters, and these include dopamine and serotonin. So the end of these synapses releases transmitter into the synaptic cleft to communicate with the next neuron, and then that neurochemical is rapidly removed from the synapse by having an active pump, an uptake transporter, pull it back inside the cell to allow the next message to come across. So that's called uptake. Moving up to a different level of analysis. A moment ago we were talking about what's happening at the level of the synapse, okay? But the other thing that we have to think about is what's happening at the level of systems. One of the main transmitter systems in the brain, called the dopamine system, is very important for determining motivational level of arousal. It influences memory and learning. It has a broad effect on brain function and behavior. And many of the drugs of abuse have either a direct or indirect effect on that pathway in the brain. So the way in which cocaine works is by blocking an uptake transporter, okay? A dopamine transporter, taking things from the outside of the cell to the inside. Cocaine prolongs the action of dopamine by inhibiting its clearance from the synapse. Therefore, more dopamine is floating around, and able to act as a message for a longer period of time. And so what's the consequence of this? Let's just take behaviors like movement, locomotor movement, or just motor agitation, moving around. As you increase the level of dopamine that's floating around between the synapses, the intensity of the motor activity becomes greater and greater and greater. It also starts to become more focused. Okay, so the range of behaviors narrows as you increase the amount of drug, and the intensity increases. And these behaviors become a repetition of the same thing, a stereotype behavior. And it's a behavior that doesn't look as though it has any meaning in the world. I mean, it might be, twiddling with a knob or searching endlessly for an imagined lost object. And the amount of repetition is in direct proportion to the amount of dopamine that's in the space between the neurons. So crystal meth is a derivative of amphetamine. Again, going back to mechanisms of action, we said that cocaine blocks this uptake mechanism that takes the dopamine from outside back in. So what amphetamine does is it blocks that uptake transporter, but it does something else. In the presence of the amphetamine, there's a buildup of free dopamine in the terminal. And then this protein that is normally bringing molecules from outside to the inside, that uptake transporter, amphetamine turns it round so it takes things from the inside outside. So now what you're getting is an increased release and then blocking the uptake. So this is a very powerful way of dumping out a lot of dopamine. Cocaine has to wait for something else to release the dopamine. It doesn't release it. It only blocks reuptake. Whereas crystal meth loads up the terminal, turns around the conveyor belt, and instead of having the conveyor belt bring things from the outside to the inside, it turns it around and it conveys all this buildup of dopamine directly out of the cell. So you get these huge buildup of transmitter. The other component of methamphetamine is its toxicity. Low doses of these drugs actually can be beneficial to thought. There's no question about it. A small amount of amphetamine used discreetly can focus your attention, but we're talking about excessively high levels of these drugs, which then lead to racing of thought, an inability to focus, and then similarly to becoming, agitated. And so studies have looked at the effect of high doses of both amphetamine and methamphetamine on brain function. And there's clear evidence that both of them, but crystal meth in particular, can produce clear, irrefutable loss of brain neurons. So you're chemically zapping neurons in your brain if you use too much of this drug too long. So the action of heroin on the brain is really quite complex. It involves two separate systems. The first system it shares in common with cocaine because it increases the amount of dopamine that's available in the synaptic cleft. But cocaine does this simply by blocking the reuptake process whereby dopamine is cleared from the synapse. Heroin works on the other end of the neuron near the cell body. Now, the action of heroin is not on the dopamine system itself, but is on an interneuron, which is normally inhibiting the electrical activity of the dopamine neuron. So heroin, by binding to opiate receptors on the interneuron, removes the brake on the dopamine system and thereby increases the firing of the cell, causing an increased release in transmitter. Let's remind ourselves that heroin is a chemical that's derived from a plant, the opium poppy. The way in which this was all discovered is people obviously knew for 5,000 years that opium affects the brain. So then in the mid-1970s, it suddenly occurred to somebody that there might be a specialized receptor in the brain that was designed to grab a hold of heroin. And then they found that. And once that light bulb went off, they said, hang on a second, why would these receptors be there if the brain itself wasn't manufacturing something that resembled the compound that we get from opium poppies? So then they went looking in the brain and found the endorphins. And this system became known as the endogenous opiate systems of the brain. Now, the normal physiological function of these systems is to provide emotional states that relieve pain, and they're also activated to produce feelings of well-being and happiness. So in the case of, say, cocaine and methamphetamine, it's the dopamine that's doing the work, not the cocaine or the amphetamine. Whereas in the case of heroin, it's heroin itself acting directly upon specialized receptors in the brain that somehow or other leads to the feeling of well-being and euphoria that defines a rush. So it has a 1-2 punch. It can amplify the activity of the dopamine system and then convey its own message. And this is probably why it's so powerfully addictive. Because heroin can influence two different systems, the dopamine system and then directly its own opiate system, it's not surprising that it can give rise to different states. Its initial action is actually depressant, and it causes a decrease in activity and can even lead to sleep. But this effect wears off quite quickly and is followed by the stimulant action of the drug, presumably through its action on the dopamine systems. And so you get initial inhibition followed by excitation. And then the next thing, which is really important, is that the ratio of the inhibition to excitation changes the more experienced you are with using heroin. So the inhibitory phase shortens and the stimulatory phase can increase. So it can sort of move the setting between inhibition and excitation. Again, what's going to happen is that the depressant effect is not going to go away absolutely, but the intensity of it and the duration of it will change. And so it can be just a passing phase, and you go more quickly into the tweaky phase with experience, as you become tolerant to one effect and sensitized to the other. Not only do opiates have these complex actions within the brain, but they also have actions at many other levels of physiology. Within the brainstem itself, high levels of opiates can lead to respiratory depression, which in turn can lead to death. And as we move into the cardiovascular system, they can alter the vascular tone of the arteries and veins and change blood pressure. And then as we descend into the gastrointestinal tract, they can influence the motility of the smooth muscles of the gut and thereby influence that physiological function. Addiction is a complex disorder that is determined by many factors. But at its most fundamental level, it arises out of significant changes in the neurochemical functioning of the brain. The assumption is that the brain is modified by repeated exposure to these powerful drugs. What I consider to be the most sophisticated theories of neurobiology of addiction right now are the ones that are focusing on the synapses, trying to understand how modification of the synapse is capturing, in essence, the memory that's left behind by the drug. There are individual differences. Some people, fortunately, can experiment with drugs and then back away. Their brains presumably aren't modified in a permanent way that would change their behavior. Other people, there may be genetic components. They may be more vulnerable. Their brain is changed more dramatically by the effects of the drug. And these changes predispose that person to addiction. If you stress an individual several times, and then you test the effect of that drug on behavior against someone who hasn't been stressed, the magnitude of effect of the drug is greatly increased by the prior experience of stress. And if you are unfortunate enough to have several stressful experiences early in your life, and then you encounter a drug later in your life, you'll have a larger and more significant response to that drug than somebody who's had a fairly benign life experience without the stress. One of the things that I would bring in here is a very renowned neuroscientist at McGill, Michael Meaney. He started out studying the effects of stress hormones on behavior. These are animal studies with rats. And what he discovered was that there's a certain number of rat mothers who are wonderful mothers, and there are other mothers that are very inadequate mothers. The pups don't get groomed properly and stimulated and don't get enough nutrition. And what happens is that the poorly mothered animals are the ones that are much more vulnerable to acquiring a habit to take drugs than the animals that are properly mothered and nurtured. So that's really interesting. But what he's discovered recently is that behavior, mother-to-child behavior, has the ability to determine which genes are turned on. And the genes that are turned on when the mothering is inadequate are the stress vulnerability genes. So you have, these stress hormones are major determinants of the way in which our brain chemistry reacts. And one of the consequences is the dopamine system, serotonin systems are activated differentially under the influence of these stress hormones. We're totally ignorant about addiction, really. We're just beginning to understand it. It's one of the most complex questions in public health and indeed in neurobiology. You can identify predisposing psychosocial life events that might be correlated with addiction, and that's very important knowledge. But to assume that you're going to be able to understand and eventually treat addiction solely on that level, I think is an oversimplification, as is the assumption that if you know everything there is to know about brain chemistry, you can come up with an effective cure for addiction. I think if you understand, like in the case I was just describing, how life events influence stress hormones, how stress hormones affect the brain circuits involved in addiction, that will give you a more complete picture than just one side of the coin alone.